In their investigations, the researchers discovered that each of the 3 mutations individually have little effect, but when all 3 mutations are present, the panicles of the rice plant retain more of their seeds, resulting in a greater crop yield. It is believed that the domestication of wild rice began when our ancestors discovered and started to cultivate rice plants that do not drop their seeds easily, paving the way for stable rice production. This discovery was made by an international collaboration which included researchers from Kobe University's Graduate School of Agriculture, Japan, the National Institute of Genetics, Japan, University College London, UK, the University of Warwick, UK, Yezin Agricultural University, Myanmar, and these research findings were published online in the Proceedings of the National Academy of Sciences of the United States of America, PNAS, on June 23, JST. Main points: the researchers discovered the causal mutation in the QSH3 gene that is necessary to prevent rice seeds from falling, referred to as seed shattering. In the QSH3 gene mutation, there is a single nucleotide substitution on the gene, YABI. This mutation is found in the vast majority of cultivars for the world's most widely farmed rice species, Indica and Japonica. The researchers found that plants with only the QSH3 gene mutation drop their seeds naturally. Seeds still fell with the QSH3 and SHISH4 mutation-mediated partial inhibition of the abscission layer. However, the addition of a mutation at SPR3 that causes closed panicle structure resulted in the majority of seeds remaining on the plant, thus increasing the crop yield. An analysis of structural mechanics was performed to determine the relationship between panicle opening and closing and inhibition of the abscission layer. The results showed that with all 3 mutations present, shattering was suppressed and the seeds remained stably attached to the panicles. It is thought that our hunter-gatherer ancestors happened to observe the visual characteristics, i.e. it is known to have originated from the wild rice weed Oryza rufipogon. It is believed that rice began to be cultivated when hunter-gatherers in ancient times chose individual wild rice plants that had suitable characteristics for this purpose. However, the current research team showed that this SHISH4 mutation alone is insufficient to prevent seed shattering loss, suggesting that other gene mutations are also involved. With a focus on the early history of rice cultivation, this study brought together specialists in plant genetics, archaeobotany and structural mechanics to elucidate how increasing yields of rice came to be cultivated. Research methodology: seed shattering is caused by a structure called the abscission layer that is formed at the base of each rice seed. The researchers found that a single nucleotide substitution, from cytosine to thymine, in the DNA of the QSH3 gene is required to inhibit the abscission layer, in addition to the aforementioned SHISH4 gene mutation. This QSH3 gene mutation is found in the main types of rice that is cultivated worldwide, Indica and Japonica. Individual mutations related to seed shattering, for example in genes SHISH4 and QSH3, cannot prevent shattering in wild rice plants on their own. However, the researchers discovered that when SHISH4 and QSH3 mutations were combined, this partially inhibited the formation of the abscission layer, which is required for seed shattering. Despite this, they concluded that such a small inhibition would not be enough to produce a stable crop yield, as seeds drop easily in a natural environment. Thus, they decided to focus on panicle shape next. Panicle refers to the clusters of thin branches at the top of the rice plant that carry the seeds. Wild rice has an open panicle structure which enables the seeds to fall easily. They then investigated the yield of each plant. They found that individual mutations had little effect and that even combining two mutations did not result in a large yield increase. However, when all three gene mutations were present, the yield increased exponentially. An analysis of the structural mechanics of the closed panicle alteration and the abscission layer inhibition revealed a complementary relationship between the two. The burden of gravity on the seed bases abscission layer is lower in closed panicle plants than in open panicle plants, which potentially brings about an even greater crop yield by further reducing seed shattering. However, a bundle of 3 arrows is stronger, and by showing his sons that 3 arrows together could not be broken, he explained that the 3 of them should work together govern the land. Rice has been a source of daily energy for people for thousands of years, and some Japanese rice cultivars are considered cultural works of art. These research results not only reveal the seed shattering mechanism, they also give us insight into the long history behind the improvement of rice growing. Thanks for watching. Please subscribe my channel for more videos.